Afternoon, and thank you everybody for joining us for this week's Internal Medicine Grand Rounds. It's my pleasure to introduce Dr. Kenneth Remy, who will be our speaker today for Grand Rounds. Dr. Remy is a tenure-track associate professor and NIH-funded physician scientist in the departments of internal medicine, pediatrics, pathology, and biochemistry in the divisions of pulmonary critical care medicine and pediatric critical care medicine. He serves as the director of the Division of Pulmonary Critical Care Medicine Basic Science and Translational Critical Care Research and co-director for Clinical Basic Science and Translational Critical Care Research in the Division of Pediatric Critical Care Medicine. He is the inaugural director of the Case Western Reserve University and University Hospitals of Cleveland Blood, Heart, Lung, and Immunology Research Center, which launched in spring of 2023. He is also the Ellery Sedgwick Jr. Chair and Distinguished Scientist in Cardiovascular Research. Previously, Dr. Remy was an associate professor at Washington University in St. Louis in the departments of pediatrics and internal medicine. At Washington University, Dr. Remy successfully completed the Academic Medical Leadership Program for physicians and scientists in the School of Medicine, the Olin Business School, and BJC Healthcare. Dr. Remy's expertise lies in adult and pediatric sepsis, COVID disease, and global health. As an adult and pediatric critical care physician, his laboratory is focused on two areas: heme-based trafficking and signaling and immune dysregulation in context of diseases of intravascular hemolysis, including COVID, sepsis, malaria, sickle cell disease, thalassemia, and after red blood cell transfusion, and real-time immunophenotyping of pro- and hypoinflammatory states to identify timing for immunoadjuvant therapies. He has over 120 peer-reviewed publications and an H-index of 35. Thank you so much for joining us, Dr. Remy.
And I was a resident here some 17, 18 years ago in those seats. All right, we're going to talk today about how to make precision medicine efforts more precise in sepsis. And this is sort of a smorgasbord talk because we're going to talk about different ways that we're going to get to that end result. Okay, I have no financial disclosures. I have a patent pending for whole blood ELISpot. I'm a member of Immune Functional Diagnostics. I don't receive any financial compensation.
Turn your volume down.
And I'm a member of the Surviving Sepsis campaign. So sepsis, it's a serious threat to public health domestically and worldwide, with the United States about 270,000 individuals and 11 million deaths annually. It accounts for about 1/5 of all US hospital admissions, but isn't a factor in about 50% of all US hospital deaths. It's a leading cause of US hospital readmissions. And although our mortality has decreased over the past decade, it still remains about 20 to 25%. And for those in the audience, you know that a pathogen induces an infection in specific individuals, causing a host response. And this includes changes as an anti-inflammatory mediator, pro-inflammatory mediation, which then leads towards endothelial dysfunction, microvascular flow redistribution, and oxygen delivery alterations, inhibition of fibrinolysis, and coagulation. And during this talk, sadly, about 1 person every 3 seconds will die from sepsis worldwide. And this is what happens to our hospitalizations over the past 15 years. You can see that our mortality, although has decreased initially, and that was with the advent of our supportive care bundles with surviving sepsis, early antibiotics, and some early fluid and early prompt removal of source. We've sort of plateaued. We're hovering in and around about 17 to 20% for individuals to die. And if you think about sepsis, it's a clinical syndrome, right? You've got septic shock and severe sepsis. It's defined specifically by the systemic inflammatory response syndrome and organ dysfunction, which is what we revised recently. And it sort of mimics a lot of other syndromes, whether it's burns or pancreatitis or trauma conditions or other infection or SIRS, and sits sort of in the middle of that Venn diagram. And when you think about how we approach diagnosis, how we approach our therapies, we don't actually tackle the biology of the disease at all. In fact, we just look at specific symptoms that are non-specific in etiology and look at non-specific markers we believe are associated with this syndrome. So why are we talking about this? Well, I already mentioned that sort of a lot of folks die from sepsis every single year. And not to scare individuals, but one of the things we don't have a lot of things coming down in the pipeline is new antimicrobials. And by 2050, many of these resistant organisms will kill more than any other disease combined. This is a real problem. And so finding better ways to repurpose existing therapies and finding better ways to develop new technologies to understand specifically the heterogeneity within the disease and syndrome is important. Because those that have sepsis have these divergent host immune responses that are maladaptive and they're muddled. Some of your cells within your body, your immune effector cells, can have this early hyperactivation, others have hypoactivation, become paralyzed, and individuals over the context of disease evolution have changes within their own cells that predominate at any given time point. And we're dealing with a syndrome that not only has a high mortality rate with limited treatment options, but unfortunately, many of these conditions that affect the immune response as well as the hematologic partition also lead towards these sort of all-encompassing muddled responses that last for long periods of time. And when we think about who dies from sepsis, historically, we used to think that people would die early in the disease course. But now we know that most of the patients that die in sepsis actually die much later. They actually survive their index hospitalization only to come back to the hospital many months later to subsequently die from that disease. Wait, but Kelly Clarkson said, what doesn't kill you makes you stronger, right? Sorry, Kelly, you're wrong. In fact, if you look here, you can see that most of these deaths persist from that index hospitalization with a second sepsis hospitalization, and many of these individuals will lose their life. And when we think about those that do survive, there's a post-sepsis syndrome. These individuals endure alterations in cardiovascular disease. They've got changes in their protein metabolism and inflammation. They retain fluid. They've got cognitive changes that persist much outside of their ICU and hospitalization. And as you can see here, weeks to months, we have this sort of late multi-organ dysfunction syndrome that persists for many, many, many periods after that hospitalization. Down in the University of Florida, Phil Efron et al. Really put together a really nice clinical trajectory of the three aspects that we see. If you don't die early with multi-organ failure, some actually have recovery, and those that live have this chronic critical illness. We see this more commonly with older adults. And these individuals have persistent kidney injury, neurocognitive decline, and also can have things like sepsis-induced myopathy. But they also have persistent inflammation in some cells, some cell types, as evidenced by different cytokines. They've got lymphopenia, which leaves them susceptible to secondary infections and other nosocomial infections. And then, as mentioned, they get cachectic from ongoing changes with metabolism and catabolism. And in that CCI, which is defined as an ICU stay longer than 14 days, you can see that time to recovery is dramatically lengthened in those that have the CCI, and that many of these patients have organ dysfunction that persists. Sachin Yendi at the University of Pittsburgh tried to characterize some of these immune markers and was able to define specifically using high-sensitive CRP, interleukin-6, and then the soluble PD-L1, specifically looked at what happens over time. And many of these patients had two specific common phenotypes. Some patients had a hyperinflammatory state as evidenced by their high-sensitive CRP that remained elevated for many, many months after their index hospitalization. Others had soluble PD-L1 that actually was dramatically reduced. And so he defined this hypo and hyperinflammatory condition that persisted and to actually follow these patients for their trajectory after hospitalization. Those that had the persistent hypoinflammation had worsened outcomes over time. The problem is that these non-descriptive markers or biomarkers, if you will, are really challenging to be able to apply to patients while they're in the hospital to define specifically who you can intervene upon to mitigate these long-term consequences. And so we're always left with who dies, who lives, and who lives, sadly, to die, because they're going to come back. And so how can you understand this when an insult like sepsis ensues? And furthermore, how can we translate this information that's been done at the population level down to the individual patient that's sitting at the bedside? So why does this matter? Well, how do we treat sepsis today in 2024? Early recognition, correct antibiotics, rapid fluid resuscitation, judicious use of vasopressors, and prompt removal of sorps. supportive care. It's not directed. It's not targeted. It's a one size fits all to everyone using broad spectrum therapies, and then just assuming that we hopefully will get the desired outcome of just saving someone's life. And we do know that aspects of this are certainly important. So there are benefits of starting appropriate antibiotic therapy. This is a meta-analysis that was performed from 1962 all the way down to 2003, looking at early starting antibiotics on patients that have sepsis. And not surprisingly, those that actually got antibiotics, especially early antibiotics, as you can see here from the no effect line, actually had better outcomes. And those that got antibiotics even earlier had even better outcomes, regardless of the source of infection. And then this is the Sentinel article that everyone that joins me in the ICU should know. This is the Kumar article that came out in 2006 that was done specifically at looking at delays in antimicrobial therapy, increased mortality in septic shock. For every single hour you delay, you increase mortality from time of hypotension by 7.6%. So at least these therapeutic options probably are what reduced our mortality down from the 30s down to about 20%. But we're still not where we want to be because we're still applying these broad-based therapies. So are there specific targeted therapies? So this is a slide from Chuck Natanson on looking at human clinical trials of anti-inflammatory therapies. These were done in patient populations that all met the criteria for clinical sepsis. And you can see here at the no effect line that many of these studies, although they cross the number one here, so therefore they're going to be statistically insignificant from some perspective, but many of them actually had benefit. And to be able to tease out this benefit in this trial design, you'd almost have to have 10,000 patients in each of these trials to be able to demonstrate efficacy. And that's a problem, because we certainly don't have the ability to garner in each of these trials a couple 100,000 patients in a meaningful trial that'll get done in a timely fashion. Now, I say this because we're using these non-specific outcome variables by enrolling patients with a heterogeneous syndrome. I've said this before, I'll say it again. So when I have a headache at home and my wife has a headache at home, she takes ibuprofen, I take Tylenol. If we did a study, we'd have 50% efficacy because one doesn't work for me and one works for her, regardless if the cause of if her headache is because of me. We won't get there. But more importantly, those drugs would never make it to trial, right? That's what most of these studies are, is that they're using non-specific definitions and not understanding specifically the cause or the biology of the syndrome. And when we look at other anti-inflammatory agents and studies, you can see that they're sadly rife with lots of different problems with heterogeneity of the populations included. But wait, we went to clinical trial, we passed phase 1 and phase 2 studies, so clearly they have safety, and we're trying to prove efficacy, and they all had preclinical studies that demonstrated efficacy. But why? Well, here's the animal studies, and this is a slide from Chuck Natanson. And you can see at the top above that line 1 on the y-axis that favors therapy, below that y-axis favors control. And if your control odds of dying in your animal studies were above 1, your therapy actually was beneficial. And then if you look at this beneficial group compared to the harm, look where the human studies sit. It's like flipping a coin. They're not sick enough because you've diluted your denominator by all of the patients that meet sepsis, instead of maybe narrowly focusing in those that actually have shock or have multiple organs that are not functioning appropriately. And when you look at a number of these prospective animal studies and compare them by animal type, same way, and by rats and dogs and patients, you see the same phenomenon. We're not able to replicate the populations in the clinical trials to what our preclinical models were designed for. So why is this important, and why am I talking about it today? Well, this is a heterogeneous syndrome. You'll hear this a lot, needing enrichment, because 49 million people worldwide are affected, causing way more than $40 billion just in the United States alone. It's the #1 cause of hospitalization, the #1 cost, and 20% of sepsis patients are rehospitalized within 30 days. It's a significant amount. But when you look here at how much we spend, and this is 2011, and it's actually worse in 2020, look where sepsis sits for funding. not asking you to open your checkbooks today, but if you want to, I'll be outside afterwards. But you can see certainly sepsis is not funded in the same way that all of the other diseases there that are listed. So this is why we're talking about precision medicine. So where are we today? Where are we going? And how do we get there? So just some definitions. Precision medicine is where we're trying to optimize for genetics, lifestyle, and environment. But personalized medicine is really probably what we're trying to get at, where we're starting to prioritize our treatments to the needs and preference of the individual patient. And we use endotypes, which are biologically based, versus phenotypes that are clinically based. And we use biomarkers to help to define some of that endotype generation. Theranostics allow us the ability to use these biomarkers in both a diagnostic and potentially as an efficacy outcome variable for therapies. And then we'll talk about a few omics that are currently being used. So in critical care, in the 1950s, all the way through about 1980s, we were coming from a period of polio. We were coming to really just begin our field of critical care medicine. And this is where we started to put folks into ICU monitored settings with continued vital signs. We were supporting organs with things like ventilators that were being used more frequently. We've got advanced hemodynamic monitoring, and we first started to describe ARDS and some other syndromes that are commonly described every single day in our ICU. And as these foundations were being laid in critical care medicine, we started to then accelerate on some of the ways that we actually use these clinical monitoring tools at the bedside. We started to develop scoring systems like the Apache score and SAPS and SOFA and Muse scores to try to define severity of illness. And now we're in a new age. And in fact, probably COVID helped us because COVID allowed us the ability for multiple teams to come together to specifically look at different omics and different biomarkers that could at least inform us for certain patient populations. And so that's where we're currently right now. We're just in the infancy, in my view, for critical care medicine, precision medicine. So what's the holy grail test specifically for what we're trying to get accomplished? We want to have a test that actually tells us what the diagnosis is, identify our severity of illness, and determine the likelihood that this person's going to deteriorate so that you can intervene earlier. I want to know if he's going to deteriorate and you're not going to, because I want to spend more resources early to make sure he doesn't deteriorate when I know that you're going to be fine. And so if I've got that holy grail test, I could hopefully be able to do that. But that assumes that I can get results that turn around quickly. A lot of the stuff that we do at the bench sadly takes weeks to longer to be able to come back to the bedside, which has been hindering the way that we approach these patients in a lot of our sepsis studies. So one of the things that we struggle with is, do they even have sepsis? Is there a yes, no? Are we pregnant or not pregnant? Right? We don't have that for sepsis, which has also been challenging because many of our patients are either called pneumonia or UTI who are septic. And then other patients we are ruling out for sepsis, but they have a post-operative fever. And so it's really challenging to determine whether or not the person has an infection and are they really sick. And so there are some tools that have helped us recently to try to stratify infectious severity by high risk for sepsis and mid and low risk. And two new technologies use transcriptomic approaches. And I'm going to just briefly mention them. But the problem about rapid diagnosis in sepsis has been these low sensitivity and specificity of diagnostic options. The fact that if we use blood cultures in real time, many of them, in fact, less than 1/3 or even positive for most of our undifferentiated patients, and they're prone certainly to false positives and false negatives. And the turnaround time is certainly 48 hours. And then this all puts us down to the point that we can't delay patients with sepsis with their antimicrobials with the current level of tests that we have to decipher yes or no for sepsis, because if they're in shock, every hour we delay early broad spectrum antibiotics increases mortality by 7.7%. So Inflammatix is a company out in California that developed a 29 messenger RNA transcriptomic metrics and developed a score in and around these 29 messenger RNA transcriptomics. And so they've conducted a few tests, and their test they say can actually help you determine if you've got high risk for sepsis versus no sepsis. That's kind of where they started at. And you can see here in this B figure that they were able to discriminate with their what they call inflammatics or IMX severity score, that their ROC curve was about 0.8%. So there's some reliability there. It was better than procalcitonin, better than IL-6, and certainly better than SOFA scores and white blood cell counts alone. So it has some promise, and they put this into a large single center ICU study of 200 patients, and were able to find that discrimination. Furthermore, they moved into the emergency room department to try to see if they can deploy this test earlier. And in 6 emergency departments with a little under 400 patients and collecting at 24 hours after their sepsis diagnosis for these 29 messenger RNAs, looking at 28-day mortality, they were actually able to have a relatively decent specificity and sensitivity for sepsis. So this right now is currently being developed for FDA approval, and they just actually got their first FDA letter, and they're hoping that they can actually get this into patients soon. Their competitor, Septisite, which we've actually demoed here at our institution last year, uses 4 different markers. They use 2 specific white blood cell genes, and then they get 4 different signatures in their mRNA in white blood cells. And from there, they're able to decipher whether or not they believe there's an infection or no infection. And now they're able to, based off their 4 patterns of these markers, to determine whether they believe it's bacterial versus viral. This is certainly in its infancy, but it is actually now FDA cleared as a rapid host response test for sepsis. And they also have a Sepsis score, and they get results in less than 60 minutes. We actually demoed it here. Our results were not nearly as good as the company's. So I think that there's an opportunity to certainly refine the population that we would be using such a test. But their AUC was about 0.85 for being able to adjudicate against clinical sepsis, those that have sepsis. Furthermore, they compared it to procalcitonin as well as in use of lactate. And you can see here in red, that's their Sepsis score, and it certainly did better, at least, than the test that we use frequently. So there could be some promise, perhaps, in time with these tests. But some of the problems still are that although this one comes back in 60 minutes, the other test certainly takes a lot longer. And we're still trying to understand, does it work in the same way for those that have postoperative infection in the surgical world? What about those that have less white blood cells because they're immunocompromised specifically from their chemotherapy drugs? So they certainly have to be at least evaluated in bigger studies. But hopefully that's one way that we can get to a more precise way to diagnose the Sepsis syndrome. Does it matter if we know the name of the bug? Well, that's really interesting, right? How many other syndromes exist where you've got gram-negative and gram-positive bacteria that all get to a common pathway to one single disease or syndrome? I don't believe there is such a syndrome. I'm using this facetiously. I think that there's actually probably 30 plus diseases that make up sepsis, and we're just at its infancy to be able to discriminate between those. But when you think about this final common pathway, ironically, most of the literature in sepsis actually has pretty similar outcomes, whether you're culture positive or culture negative. So at least at face value, it doesn't appear like whether you've got severe sepsis or septic shock, that perhaps the growing of the bug by itself may not actually be as important, perhaps, as some of the supportive therapies that are provided here. But that's not totally true. There is some evidence actually that the pathogen does matter. And if you look here at MSSA versus MRSA versus Campylobacter, and certainly when you get down to things like KPC, there are differences in mortality, especially in these organisms. And if you look at their crude versus attributable mortality. And so finding better ways to actually define these bacteria quicker, including things that are precision medicine, if you will, like matrix-assisted laser desorption ionized time-of-flight mass spectrum. I call it MALDI-TOF. But it's one way that people are trying to get towards the name of the bug and perhaps its resistance patterns quicker so that the appropriate microbials could be deployed faster. What about moving from diagnosis to disease? Well, I run a laboratory that uses mice and uses human samples. We talked a little bit about the discrepancy between humans and some of the animal studies that have been done for some of the therapies for sepsis. So one of the problems is that animals are really easy to manipulate, but we always usually do the same experiments. But what I can't do is I can't get my mice, so I can, but we don't. I don't give them beer. I don't give them actual cigarettes. I don't give all the environmental things that certainly confound our patient population. I can alter at least their ages, but not exactly the same way that we see all the patients coming through the doors in our ICU. And I can't mimic this. And if you look at this picture, all of those individuals look different, but they're not treated differently in most of our current sepsis studies. We actually don't specifically stratify patients by sex or by ethnic background, or perhaps changes in their genome or their epigenomic data. And so we can do that in our animals and actually be able to get to mechanism and be able to target that. But in the current system that we work in, we're not doing a great job specifically in our clinical trials. So how do you actually get there? Well, how do you find precision in a syndrome? Well, Chris Seymour and group have developed some clinical phenotypes, as you see here, alpha, beta, gamma, and delta. And they've actually demonstrated differences in 28-day mortality, but these are still very non-specific. And how can we figure out why this patient got this disease at this time, and why did someone else have that different outcome? Well, here's one of the fundamental problems. First of all, you can see here in scenario 1, very simple, when there's no multimorbidity with a patient. They have no other problems. They're healthy, they get an infection, and you can see that every cluster, if you had the exact same patients in a study, would have the same set of comorbidities. But what happens when you start to have multimorbidities? You can start to see specifically comorbid conditions can help us actually look at discrete groups. But then we start to have a lot of different discrepancy based off of timing of some of the patients that come in with those multiple morbidities. And it makes it exceedingly challenging to be able to actually do clinical trials with sepsis. And then furthermore, what about those that come back a second time and have post-sepsis multimorbidities? What if the therapies we're doing for our patients that are helping them in the short term may in fact also have iatrogenic effects, providing new multimorbidities? And that's where this gets really challenging. And then furthermore, how do we generate real-time data? So Peter Hovmand here is helping us with our new center. And if I've got time, I'm going to tell you a little bit about it. But we started to development of a new center based off of sepsis with a number of different disciplines in our heart, lung, blood, and immunology research center. And you can start to see in this, there's so many different decision points and different aspects of sepsis that make it very complex to really try to understand how to take meaningful data and package it in a way that if you do something here, it's going to affect something over here, because there's so many branch points of causality. The other thing we're faced with in the ICU is that we are inundated with data. There's the volume, the quantity, the dimensionality of our data, the speed at which it comes in, and that's only at a static time point. But our patients are dynamic and they change. And so we've got lots of different data points that make this challenging to be able to take all of that data and then immediately be able to spit it into something that's going to give us something meaningful at the back end. And so how to analyze our data quickly to be able to use it quickly has been a problem. And then taking static from continuous monitoring data, at least in real time, has been a challenge for us. But there's great things certainly that we have at our disposal that we're still trying to learn how to use, like my wearable technology. How do I look at heart rate variability and temperature changes with my wearable technology for those that get discharged with their primary index hospitalization for sepsis in a fashion that can tell me who's at risk to come back? And if I can mitigate some of those symptoms earlier, maybe we can save their life quicker. And some of the other imaging modalities. And then omics. Everything is an omic. Genomic, proteomic, physiomic, transcriptomic, metabolomic, exosome, epigenomics. And then we've got other assays that we certainly also evaluate, right? All of the different classic flow cytometry, the gold standard by which we actually conduct most of our studies in sepsis for precision medicine. But you've got these other tests that certainly are really important to understand alterations in both RNA and DNA, and then protein expression, and who's at risk and who's got a higher chance of actually decompensating, and could I actually change or target that specific pathway so that we can take that downstream effect and improve it. I always say I'm one heat map away from cutting my ear off, because lots of papers now are inundated. How many people carefully read through all of these heat maps and say, I exactly understand what's going on here? It's challenging. Most of the data that most of us that are collecting we actually send out for other people to analyze to some degree because it's just too much volume for us to be able to look at most of these pieces, but we get a pretty heat map at the end. But what does it all mean? And that's the fundamental question with all of the data that we're exposed to, because there's challenges of scale in our precision medicine approaches. So if you think about that, a single cell sequencing in an individual patient, just a single cell, will give you hundreds to low thousands of pieces of information. But what happens when you start to sample multiple organs in a single patient? You start to get tens of thousands to about 100,000 pieces of data. And then what happens when you sequence across millions of patients and at millions of time points? And what does it all mean at the individual level and at the population level? So what are these tests that we talk about? So if we think about metabolomics and cytomics and proteomics and transcriptomics, there's a lot of different things that we certainly have at our disposal. So one of the things that most of us at least use descriptively is using ELISA for cytokine quantification. This was not meant to actually hone in on specifically this, to just demonstrate that many of us will use cytokine quantification at single time points to see if our downstream effects for TNF alpha, IL-1 beta, IL-6, IL-10, interferon gamma, if it increases or decreases, what happens when you put a positive or negative stimulus against that? And does that actually inform us at that time point whether the patient is hyperinflamed or hypoinflamed? And so this has been historically how we've all evaluated patients with sepsis, because we would like to think that certainly fever, IL-1 beta increases, TNF alpha increases are certainly reflective of a hyperinflammatory or a cytokine storm, if you will, in patients. And there's some good data from Mark Hall and others that demonstrate that if your TNF alpha response is blunted from lipopolysaccharide, so you don't get that whopping increase, but in fact your cells don't respond, that gives you at least a marker that either monocytes or other affected immune cells in the innate system actually could be sluggish, paralyzed, or suppressed. And that's informative because that may not be the patient you want to give corticosteroids to, because making that worse may render that patient susceptible to a secondary infection or a nosocomial infection. We typically don't use that right now, but this could be one simplistic way that we might actually address some of the patient issues at the bedside with a personalized approach using an old therapy or assay like ELISA. We use ELISpot, one of the other ones that we use, which is ELISpot allows you in a 96 well plate to actually use a positive stimulus. So if I'm trying to stimulate cytokine increase numbers from the cells that are producing TNF alpha, I'll use LPS, and I'll get a number of these little spots on that little dish. And the size of the spot and the number of the spot tells me specifically that cytokine of interest and that it's being produced. And if it doesn't produce that spot, then I know that those cells are sluggish or not acting effectively. The same is true, and I use that as an innate marker. And an adaptive marker, I might consider using something like interferon gamma, where I use a positive stimulus, CD3, CD28, to try to stimulate that to see if those cells are responsive. And if they're not responsive, that also gives me that information that that may not be a patient I want to give corticosteroids. And if they are less responsive, maybe I want to stimulate them with something like GM-CSF or something like interleukin-7. And you can take cells in this platform and actually expose them to therapies. And so that's one of the things that we started to do in COVID. We demonstrated that the non-survivors in patients in their interferon gamma production had much less production after stimulation. And then from there, we actually developed therapies. And one of those therapies was interleukin-7, and another one was baricitinib. And we actually did this in a dish in St. Louis. And we're able to refine the populations to some degree to be able to differentiate who those patients that might not benefit from anti-inflammatory therapies, but could benefit potentially from pro-inflammatory therapies or restorative therapies. And so this is just one way that you could actually take a simple technology and get a result within 24 hours that you can actually apply at the bedside. But as I mentioned, flow cytometry is the gold standard for functional immunophenotyping. And so we use this. It measures properties of cells as they flow in a fluid suspension across an illuminated light path. And as you can use this for quantitative and qualitative analysis of several properties of different cell populations. You can understand the particle size, granularity, relative fluorescence intensity, and you can understand in a single antibody response or in multiple antibody responses. And then this is used for cell sorting and cell proliferation, cell cycle analysis, DNA content, apoptosis, and immunophenotyping. And much of the work that was done by Volk and then Monterey in flow cytometry in sepsis has actually demonstrated specifically things like HLA-DR decrease in those that actually have a high propensity for death. And you can see that these were linked for most of them, like CD3, CD8 total lymphocyte counts with death. So one option. The challenge is that flow cytometry takes a bit of time to be able actually to use and be able to integrate into your clinical practice. But as I mentioned, those original studies in 1997 with Volk actually spurred a lot of folks since then to use HLA-DR specifically as an analyte in sepsis. But what we haven't really done well is used it in a way to actually refine our clinical trials based off your response in HLA-DR. But one opportunity for the future. And the other thing that we don't use flow cytometry for is we like to see those that are positive. But what we actually failed to miss is actually revealing the dim populations or events that are actually in the background, because those are the ones that perhaps may actually be beneficial to actually start to find putative mechanisms to modulate. And then we can certainly move away from single and look at multi-parametric evaluations with flow cytometry and look at different parameters to actually refine some of these immunophenotyping techniques with high-dimensional multi-parametric flow cytometry. And these are just some different ways that you can get multiple time points. Once again, what does it all mean? What's the context in the patients? We're still just tackling this at the beginning of its infancy in our field for critical care, but certainly utilization of this has got great promise. And then undeniably, our ability to use other omic evaluations, whether it's use of proteomics, which is what our lab uses, cytoph or cytometry by time of flight, where we actually use, instead of fluorescence, we actually use mass spectrometry and specifically label with metal isotopes instead of fluorophores. And we've used this actually in our work in COVID. So in St. Louis, we were one of the first groups to use cytoph proteomic evaluations of patients with moderate to severe COVID-19. And with this, we were able to look at 75 features per sample, and we looked at 5 different cell populations and unearthed that STAT3 was the predominant signaling pathway activated by COVID-19, and then subsequently took this information to the FDA and the NIH, and baricitinib was added to the armamentarium from us and John Weary's group at Penn and another group that actually recapitulated similar findings. It actually was able to be used in a fashion, at least for populations, to at least evaluate patients with sepsis that perhaps have alterations in their STAT3. And so it helps refine some of your populations that way. And so genomic and transcriptomics, so people know a lot about single cell RNA sequencing and transcriptomics, we use nanostring. This is a lot of the different data points that you can see in this one, yet again, heat map in the center of the screen. But you can start to look at specifically genomic RNA sequencing to actually refine those populations, so then you can start to modulate just those and those that have altered expression. And we use also nanostring, which is a transcriptomic approach, where we can look at 800 targets specific to an inflammatory condition that have been carefully curated specifically for that disease. That way we can get our results back in less than 24 hours. And we've been fortunate specifically in our sepsis patients and that this is an area that we're hoping to use because it's got more ease to be able to refine and get the data analyzed in a quicker fashion because you've got less targets than you might see in a complete whole genome or in single cell RNA sequencing. And so where are we at? So we're trying to really refine how to approach the pathogen and how to enrich the host, both in understanding their disease and refining the population for clinical studies. And as I mentioned, we've had this one size fits all with sepsis as a syndrome. And we need to start to really look into individual patient fingerprints of the biology or functional status in real time. And so a number of folks have sort of thought about this theoretically. And I've said this a few times, we're only at the bottom of the staircase. We're still just in this part of just really parsing out what might be the best assays to be able to be used in clinical practice from the bench back to the bedside. And how can we use these to get to where we want to be? Because if we can start to use biologic phenotyping or endotyping in a robust response, we can start to then pattern our RCTs to be able to be used in populations that they would be best served. And if we go back to this, instead of needing 10,000 patients, perhaps you only need 500 patients to be able to develop better ways to use these therapies that demonstrated safety in phase 1 and phase 2 trials. They didn't demonstrate efficacy, but they were darn close. And so you could repurpose them specifically in carefully done studies that would allow that. So Carolyn Calfee and Michael Maffei, friends at University at UCSF, had this beautiful picture in Nature Medicine in 2022 and also in 2024. And they specifically look at that heterogeneous group of syndromes, look at sampling to bring down different biological characteristics that we can collect at the ICU and in the hospitalized patients and perhaps in the future outpatients with wearable technologies, and take these different parameters with our biomarkers, including advanced omic evaluations of these patients and imaging features, and move it into a machine learning approach that affords us the ability to take all of this to be able to bring down to a point of care test in our ICU populations and hospitalized patients to be able to get towards those novel trial designs. So our group, with a number of different groups in the country, is currently doing this. We've got a large RL1 trial that's been evaluating how to stratify these immune endotypes in sepsis. But we're still stuck with trying to understand, because we all work in our little siloed areas of our own interests in specific areas. Our group looks at a lot of T and B cell adaptive changes, but other groups perhaps look at these other ones. In sepsis, what it really needs is a change in nomenclature that actually takes into account all of these different effects in each individual patient to then be able to characterize, you've got sepsis A, you've got sepsis B, you've got sepsis C, etc. And as we move towards that, we then can precisely apply therapies to each of those endotypes or disease entities that have new names now, and then develop our appropriate RCTs and adopt this into practice, and then figure out and predict who's actually going to die, who's going to live, and how we can stead off those that are living to die so they don't die. And so to get there, we certainly need a lot of smart people around the bench and clinician, including those that work in computational biology and artificial intelligence and understand social determinants effects and epigenetic effects, et cetera. And I think that's how we're finally going to get towards decreasing that 18% mortality in sepsis. And then furthermore, how to take this information and certainly help our marginalized populations and those that live in LMICs that may not have access to these technologies to be able to demonstrate what fingerprints would look like also in those populations is certainly important. And so to get there, we have to have alignment in our basic science mechanisms and our biomarker development, and then our outcome measures that reflect these biomarkers, not just as an ancillary association, but actually we look at mortality and effect on the biomarker of interest that we're using to actually gauge how to enroll people into the study to begin with. That's really important. And I think this with adaptive trial designs will get us where we want to be. So as I start to near the end of this, the Human Genome Project, many here probably know, is a large, well-organized, highly collaborative international effort, started in 1990 all the way to 2003. Included one of my mentors, Francis Collins, who was fantastic, tells great stories about this. But what's most interesting is that most of the sequences generated by this project was actually from only 11 donors, plus one, total of 12. That's it. And yet we've gotten so much information specifically from that. But I want you to just think about this. Since the conclusion of the Human Genome Project, 7 million people have died from COVID, but about 250 million people have died from sepsis. That's an astronomical number. And my belief is that we need a human sepsis project to get to try to impact and decrease the amount of death that occurs from this horrific syndrome. I said one person dies every 3.5 seconds. I've talked a lot in the last 50 minutes. And the way that we get there with this Human Genome Project is to start to bring all the disciplines to the table. And many of these won't be physicians. They're going to be people who work at Google and Meta and other groups like that. But we need to engage this, because if we can pay someone $700 million to play baseball, perhaps $200 million could actually save people's lives. And so we're actually in process of this. So I'm currently writing a grant proposal to the NIH for a meeting, and we've got a meeting with Meta in the upcoming months. So this is a good first step to get experts to the table to be able to look at all those different partitions to hopefully be able to impact and understand the biology to rapidly deploy better theranostics. So sepsis represents complicated biology, heterogeneous set of host immune, hematologic, and biologic systems responses that require further characterization into usable immune and clinical endotypes to inform therapy. We need to look at individual patient signatures to move towards that personalized medicine. We need to understand the biology of the syndrome so that we can then refine that heterogeneity of both treatment effect and diagnostic effect to understand functional immunologic, hematologic, and endotheliopathic and other tests. We need professional help, but not just doctors. Lots of different smart people in the room to help us understand the data that we've got in our own hands and how to develop new data to be able to make it more useful. And then we need an accelerated pathway to be able to initiate what I believe is the need to create that largest sepsis collaboration that brings in industry, federal funding, philanthropic, and other ways to improve our precision medicine approaches. And I think that's how we get to the point of moving the field forward. So that was the end of first talk, but I'm happy to stop here if there's questions. Yeah. Okay. Well, why don't I talk for about 6 minutes about the new center so that folks are at least aware of it, that are unaware of it so far. So really fortunate that we are embarking upon now the startup of this new blood, heart, lung, and immunology research center. And fundamental questions that help derive the development of this center were, what are the state of interdisciplinary research at our hospital and case? What areas are we hoping to enhance? Where do we want to be in 5 to 10 years? And how do we engage talent in the lab and outside the lab? And so some in this room may or may not know, there was a great paper in 2022 in June talking about the mass exodus of those at their mid-career. Funding, certainly at the basic and discovery science level, has certainly been plateaued for a long period of time, although outcome research has certainly increased. And so a number of folks are leaving this area. And so how do you find those scientists that are going to really be able to understand the biology of disease to be able to move this eventually into patients? And so there's a huge spectrum of translational research from T0 to T5. And when I arrived here, I was fortunate to have great conversations with a number of our folks here. And one of the things that certainly was in need of some additional enhancement was, how do we create a better scientific community? And from that, I was fortunate that Dr. Salat and Dr. Simon and Dr. Jane engaged in the concept of this new blood, heart, lung, and immunology research center that would expand the CVRI that both they and Sanjay Rajagopalan have done a wonderful job in being able to develop this, but broadening it to areas that really made sense to start to bring those compartments together to provide transformative leadership and research training and discovery scientists in those areas that were age agnostic, disease agnostic, but allowed for us to really be at the forefront of discovery scientists. And so in 2022, the concept of the development of New Center was born. In 2023, the expansion and application to university hospitals and case to become a type B center to expand the CVRI was approved. The JSLC approved our operating budget. In the early parts of this year, we've been working on a lot of different aspects of programming, developing a website that currently is getting ready to launch, and a phased opening approach with our first grant rounds that occurred in April. And the JSLC furthermore approved a biorepository funding with a main programming kickoff. And none of this would have ever occurred with Bob and Dan and Sanjay and Mukesh before he left, certainly, and his wisdom since then, Stan and Yi Fritz, who really is the glue for most things that occur in this realm. And so our concept was how to engage discovery scientists to be surrounded by those that do all that stuff I just talked about in sepsis, database and science informatics, AI, social determinants of health, environmental influences, predictive omics. And how can we engage those people involved in all interactions? And from that, we came up with 5 thematic areas that encompassed heart, lung, blood, and immunology research in different areas: fibrosis, hypertrophy, autophagy repair, epigenetic predictors of disease, brain organ interactions, and vascular biologic alterations, and immune monitoring and treatment. And these thematic areas will help forge groups of teams of scientists here at Case and at university hospitals to be able to take a discovery at the bench and get it into patients faster. And so our center is certainly led by Stan and Dan. I'm the center director with Dave Zaidar and Julian Stelzer as our deputy directors. Yi Fritz is our executive director. We've got a fantastic scientific advisory board and executive advisory board. And we've got thematic leads and are growing that right now. And that's in the process of growing over the next couple of weeks with leads of those thematic areas I just showed you. But this program and this new center has a scientific editor that helps people get their grants to success and their manuscripts to a higher impact journal. We'll be adding a biostatistician and a grant specialist very soon. And we have pilot funding for those that are going to be able to enhance the mission of the center with collaborative approaches that bring scientists together. And so our website should be going live soon at the end of this month and will encompass a lot of the different aspects of both growing out of the CVRI, but certainly some other new areas that bring in immunology and certainly blood research. And our membership will be open to faculty at the full member level, associate members, clinical members, and then also trainees and laboratory members, because all of these folks make up our scientific community. And we want to be able to house them in an opportunity, be able to get to the best science that we can get to. And membership allows pilot funds. If you're not a member, can't apply for one. Use of developmental funds and career development programs that we have in place. The ability to actually come to the seminars and be part of our large research days. To be able to get those scientific editing services to help you with the grammar and the language, to be able to get feedback in a mock study section. And then for our junior faculty, both at the PhD and MD level, a new career development program will begin in July. And it'll tackle a number of these topics over a 2-year period, led by a number of key scientists on campus, and really will help people to start to think about how they grow their career into the area that they want to. But many of these topics weren't taught to me in medical school, weren't taught to me, at least in training. I learned them through a number of different courses. And so we've got an opportunity where we can certainly grow a lot of the background information into our junior faculty. And then our seminar and programming includes grand rounds on the first Thursday of the month. A second Thursday is going to be our chalk talks, where people actually don't have slides, but they go to a board and they put a name on the board and they draw a figure and they say, this is what I want to study and how I'm going to study it. What do you guys think? And they get peppered with questions, but what it does is it helps refine the science. And it also finds new collaborators so that we can put in not just smaller grants, but large center grants and program grants and things that are certainly going to continue to enhance our research and our community. And then the third Thursday of the month will be our junior scientist faculty development, which is going to be open just to those individuals and mentors. And then our fourth Thursday of the month will be our technical methods discussion. I was only taught how to do these methods. I have no idea, but I see it and I read about it, but I don't understand it. We're going to have an opportunity where people can learn new techniques and find those collaborators that use those techniques so they might actually be able to broaden their science. And then the fifth Thursday of the month, if we have one, will be understanding the diversity of science and the research. Our research should also reflect the greater Northeast population that we serve. And certainly the scientists that are in our center should also reflect that population. And as the center director, I'm certainly dedicated to providing that opportunity. We had our first grand rounds and Alan Doctor talked about role of red blood cell enos and oxygen delivery homeostasis. We had about 55 people at the first one. We've got a developing relationship now with the intramural world at the NIH, where we're going to have seminars 4 times a year and talk about ways to forge intramural, extramural relationships. And then we've got $50,000 grants. We're giving 4 out this year that actually will enhance as pilot awards to those members that are part of the organization, that are part of this. It's a 4-page grant application. It's small. I don't want people to be spending 13 months to write a grant to not be able to actually get the preliminary data they need. They need a pilot award to be able to get that larger grant. And they also got to demonstrate that they work collaboratively with other aspects of people here on campus. As I mentioned, our scientific editor is going to be able to give and have study sections twice annually so that you'll get really meaningful feedback on your grant, so that you don't get that feedback from study section, but you get it from the mock study section, and instead you just get the award. And this will also help break down some of the work that people do when they write grants on a lot of the things that we're going to house internally within the center on things like facilities and environment pages and your bio sketches, and our editor will help edit those so that all you're working on for the most part is really your strategy. And then Dave Zidar is going to be hopefully giving grand rounds in the fall for this new center. And we'll talk about our new BHLRI biorepository that'll be able to take patient samples and bring them to a location that'll be coded and have data associated with IRB approved usage, and both in a de-identified or identified, depending on how you are going to access these biorepositories, and will afford us the ability, especially for clinician scientists, to be able to actually partner with bench scientists to be able to start to ask some meaningful questions. And so where we currently sit right now, we're hoping that after kickoff next month, we'll have about greater than 50 discovery scientists. That doesn't include all of the folks that work in labs. We've already recruited 4 individuals here. One of them is here today, good old James Ross from trauma surgery, and 2 are coming in July. We're also going to have scientific happy hours. One is only for lab personnel. The other one is for lab personnel and PIs, so that we can create this opportunity to actually get to know each other and understand what we're all working on and figure out, hey, I got a lot of this stuff that's expiring. Does anybody else need it? Because there's nothing worse than have a lab full of expired stuff that could have went to someone else who didn't need to spend an extra $12,000 on stuff. And so we're excited about these beginning in July. And our hope then after the first year is to have a major research day dedicated towards the mission of our center with Bill Sheeman's help and Dan Simon and Stan Gerson's. So if we start to think about UH, the entire UH, is that every hospital bed really should be a research bed. We should be learning from every single patient, and that we should really be understanding cohorts of each disease, having a research biorepository so we can improve care. We don't need to be buying all of our Jackson mice and lab when we've got people here in Northeast Ohio that reflect tremendous diversity in Parma and in our Amish communities and our Beechwood populations. We've got great epigenetic diversity here, and trying to actually leverage that to be able to use this for research, I think is tremendous for our region and actually to lead the country in this area. So we're excited about this. So I'm hoping that we're going to be building this. Always happy to have trainees come and talk about maybe areas that they can engage upon, but we've got a lot of great team members that are hopefully here to help. With that, I'll say thank you. All right, so Catherine Bruning. Are we currently under or over treating at our institution for sepsis? This is a great question, Catherine. It's hard to know because when we went back and looked at a lot of our patients using things like Septisite, there are a lot of patients we called sepsis that actually didn't meet at retrospective evaluation sepsis. So I think under those circumstances, there may be a population we're over treating. I think grossly we're probably under treating because we're usually reflecting patients as just having pneumonia and not having sepsis. And by definition, if you've got pneumonia and you have oxygen, you've got an organ failure. And so probably these patients do require earlier antibiotics and earlier manipulation. So I think we're probably under recognizing most patients with sepsis. And then as far as the next question, are the MRI measuring the host response, or it depends? So it depends on inflammatics or septicite. They're different. One is looking at the pathogen. The other one is actually looking at, I should say, the other one is actually taking in both in combination to understand whether or not it's both the host response and the bacteria. And sorry, go down to the last one here. Is it fair to assume that aging causes loss of some of the protective genes? Certainly possible, Vasu. I think that's a great point, but it's one of the reasons why we need to study sepsis, not just in isolation of our senior citizens, but we need to understand a lot of the studies with our younger folks to inform our older populations to see refractoriness of our specific immune effector cells and see whether or not there's less refractory period for certain cells in the older folks compared to the younger people. And then what are the confounding variables that make this worse, whether it's a patient with immunosuppressive and drugs on board or corticosteroids. Really, it's a area certainly ripe for investigation, but great question. Any other questions from the audience? Otherwise, thanks for joining. Really appreciate your time.